Okay, let me ask you this question. You need to answer all these questions. Okay, universal donor, universal receiver. So what will happen if a patient with blood type A receives type AB blood? Will the patient live or die? The answer is the patient will die. Second question, a patient with blood type B receives blood type O. Will the patient live or die? Live. Good. If the patient with blood type O receives blood type AB, will the patient live or die? Die. When a patient with blood type AB receives type O blood, will the patient live or die? Live. Okay. Now we want to talk about a new term, pleiotropy. What does it mean, pleiotropy? Pleiotropy means one gene affects multiple traits. One gene affects multiple traits. So far, we have been studying one gene, one trait. It's like the monohybrid cross, one gene, one trait. It is very straightforward, very simple. But in real life, there are a situation that is a lot more complicated. We call it pleiotropy. One gene affects multiple traits. An example will be sickle-cell anemia or sickle-cell disease. Why? Because it is one amino acid mutation. There's one gene mutated so that it will change the amino acids, and then it will cause two changes. One is the hemoglobin. Hemoglobin protein will change the shape, and then also the red blood cell will also change the shape into sickle shape. So these are the two changes. So one gene mutated will cause two phenotypic changes. One is the hemoglobin protein will change the structure. Then another one is the red blood cell. Originally it is biconcave disc like a donut, and then it will now become a sickle shape with jagged edge. So most of the time, people who are homozygous with sickle cell anemia, they will have sickle cell disease. They're homozygous with sickle cell allele, then they will have sickle cell disease. So the sickle cell allele is also recessive. Okay, so this is also a recessive disorder. So homozygous recessive, okay, we should emphasize this, homozygous recessive. Then they will have abnormal hemoglobin, and also the red blood cell will become sickle shape, just like this one, sickle shaped red blood cell, and it will cause all these symptoms: weakness, pain, organ damage, and paralysis. And then, well, you may call it the opposite. You may call it the opposite. We call it polygenic. You may call it polygenic is the opposite of pleiotropy. Polygenic meaning that multiple gene, two or more gene, contribute to one phenotypic trait, one phenotype. Two or more genes contribute to one phenotype. It is the opposite of pleiotropy. Okay. An example is the body height. So you may ask, well, what about achondroplasia? That is the dwarfism. Okay. Achondroplasia is the dwarfism. What I mean here, the body height, is that people like us, we don't have the achondroplasia, we don't have dwarfism, but some of us, we are taller, some of us, we are shorter. Even though we don't have the dwarfism, even though we don't have the achondroplasia, some of us may be shorter, some of us may be taller. So the body height in people who do not have achondroplasia, then it is controlled by several genes, and they are inherited separately. So as we see here, it is controlled by a great number of genes, but we just simplify here. You don't need to know too much details about this, as long as you know that okay, body height is basically a polygenic trait. And same as your skin color, so on and so forth. So we want to go back to what we briefly talked about, segregation and independent assortment a little bit. It has something to do with our chromosome, okay? As I mentioned before, genes, they are located at the specific locations. We call it loci. Loci is the plural, locus is the singular. That means a specific location on the chromosome, okay? And then meiosis causes the segregation of the two allele, okay? Meiosis causes segregation of two allele. And independent assortment really depends on the locus of the two of the allele. So I'll show you what it means. So remember Mendel's law. We have the Mendel's law of segregation. We have Mendel's law of independent assortment. We'll take a look at this. So what I mean by meiosis causes segregation. That if you remember our cell division lecture, you should understand why. But what about the locus, the loci, causes independent assortment? Then I'll explain it to you in the next few slides. So take a look. Meiosis causes causes segregation of the two allele. So so some gamete, they will get the big Y, some gamete will get a big R. As you can see that this is the parent generation. Parent generation, so they are self-fertilized, they're purebred. So all the gamete from the parent, they will have a big R and big big Y. Similarly, for this wrinkled green seed, they also have a small y and small r. Then the F1 generation will be they will have all the allele, big R, big Y, small y. Okay. So they said that the law of segregation is here. So for the F1 generation, when you go through a meiosis to form a gamete, what will happen to the gamete? Okay, some of the gamete will have the big R's. Some of the gamete will have the big R, big Y, but some of the gamete will have small r, small y. Another gamete, due to independent assortment, remember independent assortment in meiosis, this is the metaphase, this is the metaphase 1 in here. The metaphase 1 in here, it will have independent assortment, and then what will happen is that some of the gamete will have small r, big Y, or big R, small y. Link gene. Linked gene can also explain why we will have the law of independent assortment in Mendel's law. Okay. So what is linked gene? Basically, okay, the gene, they are located very close to each other. The two loci, they are very close to basically next to each other. Then we call them linked gene. So during meiosis, what happened during meiosis? So the linked genes, you remember in prophase 1, we have something called crossover or the recombination. If the genes are linked, usually during recombination or crossover, two genes, they will go to the homologous chromosome. They'll go to the other chromosome together. So when they are doing crossover, they usually go together, move together. So that's why they said that inherited as a set, and they do not follow the independent assortment. Let's see some example. So you can see that all these genes, they are on this chromosome. What do I mean by linked gene? For example, hemolytic anemia and ovarian carcinoma, ovarian cancer. These genes, they are together. They are located very close to each other with a very close loci. So we can call them the linked gene. On the other hand, for the ovarian cancer gene and let's say diabetes gene, these two, they are located far away. Then they are not linked gene. Okay, so what's the significance of this? So when the gene, they are located far away, then they follow the Mendel's law of independent assortment. They follow Mendel's law of independent assortment. It's just like the Labrador Retriever. So it's like the body color and the vision, normal or blind vision gene. They are located farther away in the chromosome. So they follow the Mendel's law of independent assortment. But if the two genes, they are located very close to each other, what will happen? Then they are called linked gene. And then they do not follow the Mendel's law of independent assortment because they usually go with each other. So when a crossover occur, when one gene jump to another chromosome, then it will usually bring the neighbor gene with itself. So for this, the linked gene does not follow Mendel's law of independent assortment. So this is the example that I just told you. So during prophase 1, crossing over, we are crossing over during prophase 1. So what happened is that gene A and gene B, as you can see, that they are closely located. They're closely located in a loci. They are close to each other. So when crossover occur, if the crossover does not occur with gene A or gene B, then they will stay together. But when crossover occur, in this case, when the gene A and gene B, they're farther away, then you can exchange the gene. Okay, gene A will do the crossover, but gene B, they will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover, but gene A will do the crossover, but gene B, they will do the crossover Oh, sorry, no. It should be opposite. Gene A will stay in the chromosome, but gene B is the one that go through the crossover. So what it means is that because gene A and gene B, they're farther away, then when there's a crossover, one gene will move to the other chromosome, but gene A will stay. So this is what we call so for this one, for this situation, it follows Mendel's laws. Of independent assortment. Okay, I just put IA, Mendel's law of independent assortment. For this one, they're the linked gene. They do not follow Mendel's law of independence assortment. Okay, how do we